My lab works on the gut microbiome, and specifically how we can look at how the microbiome affects different aspects of immunity, but also in general host physiology. So just a very brief introduction to the gut microbiome for anyone who hasn't already heard in pop media recently. There are 30 to 40 trillion microbes living inside of us in the gut all the time. They are our resident microbiota, and at 30 to 40 trillion microbial cells, they match the number of cells in the human body approximately 1:1. As you may or may not have seen in the scientific literature, numerous members of the microbiota have recently been correlated with a number of different health and disease states. Basically, pick your favorite disease, and probably the microbiome has been associated with something about that disease in one way or another. One thing that you'll perhaps appreciate about most of these studies, however, is this word correlation. So the microbiome is correlated with different disease states, but not too much has been done determining whether there's a causal relationship between these changes in the microbiome and these different disease states. So how do we go from correlation to causation, or from correlation to mechanism? Well, classically in experimental research or in biomedical research, we take an observation from the clinic, or we take a phenotype that we're interested in, and then we go in vitro or in vivo, and we try to work out whether there's a causal relationship and what the mechanism might be. There's been previously one big barrier in the microbiome field, in that the specific species of microbes that exist in the human gut microbiome are not the same as the ones that exist in the mouse microbiome. So if we want to take them into an animal model, we actually have completely different players there, at least in name. So this was kind of the first thing that my lab wanted to tackle, is how do we deal with the fact that we want to look in mouse models or in animal models at phenotypes that are relevant to human disease, but it's not the same microbes that are there. So how can we translate these findings between, in our case, mice and humans? And the answer came in just looking instead at the names of the microbes or the taxonomic classification of microbes, and instead looking at the functions that they encode. In other words, there may be different names of microbes in the human gut versus in the mouse gut, but the basic functions that they're performing and the metabolites that they're producing are largely the same. So basically, any way that we cut it, whereas there's a 3% overlap taxonomically. Oh, sorry. Whereas there's only a 3% overlap taxonomically, there's approximately an 80 to 90% overlap in terms of functions. So the answer is, okay, if we have different species present, but we know what the functionally equivalent bugs are between mice and humans, and that will allow us to experimentally investigate what's going on in our phenotypes of interest. So in order to do that, we use a method that we call functional metagenomics. So we take shotgun metagenomic sequencing of the gut microbiome. We also do the taxonomic classification that everybody else does. So we look at what species and strains are present. But more importantly, what metagenomic data gives us is whole genomic sequence information about the microbes that are present. So we know which microbial genes are present, and for those that are functionally annotated, we know what functions they encode, what kind of enzymes they encode, and presumably what kinds of metabolites they encode. So we use several different methods to get at this, including contig assembly, metagenome assembled genomes, pangenomes, and some other computational tools that have been developed in our lab. So again, what this allows us to look at is, rather than just looking at the names of the species that are present in a microbiome, the composition of the community, who's there in the ecosystem, we can actually look at the functions. This gives us access to the metabolic activity and outputs of the gut microbiome. And it's these outputs in the form of bioactive metabolites that are actually exacting effects on the host and host physiology. So how can we apply this to understanding effects on host immunity and disease? And so for the sake of time today, I'm just going to talk about one specific example and one study that we have that's hopefully about to get published. Immune checkpoint inhibitors are near and dear to my heart. As mentioned, I came from James Allison's lab, and he obviously pioneered the use of anti-CTLA-4 for cancer immunotherapy. But importantly, so immune checkpoint inhibitors harness the power of the immune system to release the brakes on the immune response to cancer and allow our body's own immune system to fight cancer and eradicate tumors. And they've actually revolutionized immunotherapy, as you can see from survival curves combining immunotherapy with different chemotherapeutics. The, I guess, most famous or predominant immune checkpoint inhibitors that are currently in use are anti-CTLA-4 and anti-PD-1. And again, they basically resulted in just phenomenal responses in some patients. So this was a patient that was examined and treated by Jed Wolchuk back before we got FDA approval for ipilimumab. And as you can see, quite rapidly, this is a patient with metastatic melanoma. I think a couple of pictures say 1,000 words. So basically, the immunotherapy took this patient that was experiencing severe lesions of melanoma and caused many of these lesions to regress within just 3 months of therapy. However, unfortunately, it's only a minority of cancer patients that actually benefit this much from cancer immunotherapy. And something that was quite quickly seen in therapy is that the efficacy of immune checkpoint blockades is highly modulated by the gut microbiome. So in the last 5 years alone, there have literally been dozens of clinical studies and papers examining the association of the gut microbiome with immunotherapy efficacy. However, again, what you'll probably notice is that each study seems to associate different species of bacteria with response or non-response to immunotherapy. So there hasn't been a consensus about which microbes are contributing to a good response to immunotherapy or a lack of response to immunotherapy. What we think about, because we think about it in terms of, again, immunology, is obviously there are some common microbiome metabolites and cell wall components that are important for modulating immune cell function. This is well known in the gut in terms of metabolites produced by the microbiome, but in general in immunology, we know that pattern recognition receptors are quite good at sensing different conserved patterns from microbes. So again, we went looking, there we go. There is precedent for this. So several other groups have looked specifically at microbial metabolites produced in the gut microbiome and how they can modulate or improve immunotherapy efficacy. And a few studies published, I think, late 2023, actually showed that the abundance of LPS in the gut microbiome corresponded to a poor prognosis or a poor response to the immunotherapy anti-PD-1. So this is important because looking at the functional annotation of the gut microbiome allows us to do is to look at some of these metabolites and which microbes are encoding the ability to produce different metabolites. So for instance, the enzymes involved in cell wall biosynthesis are actually fairly well annotated for many of the microbes in the gut microbiome. So looking at this, to further dissect this, Pushpenu Sardar, who's a lovely postdoc in the lab, examined the baseline gut microbiome composition prior to immunotherapy in a seminal study of anti-PD-1 immunotherapy in metastatic melanoma. And what he found is when he looked at the taxa or the genera of LPS-including bacteria, he didn't see good segregation between responders and non-responders. In other words, we couldn't predict who would be responsive to anti-PD-1 based on the name of the gram-negative or LPS-encoding bacteria in their guts. However, when he looked at the genes responsible for LPS biosynthesis in the gut, it actually really nicely segregated responders from non-responders and suggested that this gene right here, LPSM, was important for distinguishing between who would and would not respond to anti-PD-1. Again, importantly, we know what this gene does. So LPXM is an acyltransferase that adds a 6-acyl chain onto the lipid A molecule of LPS. And it is known that this adjusts the structure and changes the immunostimulatory capacity of lipid A and of LPS. Importantly, profiling the overall microbiome of these melanoma patients, we expanded our analyses to encompass five different data sets with 7 clinical studies from 4 different countries, so expanding beyond the initial study. And then again, applying our stringent criteria, so we only want baseline microbiomes from patients that aren't currently undergoing treatment with antibiotics or antacids that can themselves skew the microbiome. So we see that at baseline, most of the LPS biosynthesis genes that are encoded in the microbiome, of those, most of the bacteria in the gut microbiome actually encoding the ability to make pentaacylated LPS. So they don't have this ability to add the 6-acyl chain onto their lipid A. What this means is we actually have a huge heterogeneity of structural forms of LPS in the gut, and most of them are this pentaacylated LPS. Whereas LPXM, again, adds a 6-acyl chain, and LPXJ also adds a 6-acyl chain in a slightly different position. So again, though, when we look at the total abundance of all LPS biosynthesis genes in the gut microbiome, the studies that were previously published were correct, that overall total LPS corresponds to poorer response to anti-PD-1. However, when we look at the abundance of just LPXM and LPXM encoding taxa, they're significantly increased in patients that respond well to the immunotherapy. Importantly, when we expanded the analyses out to just a single study of renal cell carcinoma patients, we also see a very similar trend, that an increased abundance or increased ratio of this immunostimulatory hexaacylated LPS structure corresponds to better response to immunotherapy. Going in vitro, Satoshi Sudama, a former postdoc in the lab, looked at the ability of these different structural forms of LPS to stimulate immune responses. So using a reporter system for NF-kappa B activation, we can see that hexaacylated LPS is about 10,000 times more potent at stimulating immune responses than the pentaacylated LPS is. And similarly, in terms of cytokine secretion, so if we look at the ability of THP1 cells, so a monocyte macrophage cell line to secrete inflammatory cytokines in response to LPS, the hexaacylated LPS is much more potent at stimulating secretion of cytokine. So from there, we wanted to take this into a mouse model and look into the mechanism more deeply. And so what we did is in a implanted tumor model in mice, we have 2 different things. So we have polymyxin B. So polymyxin B is a targeted antibiotic that actually specifically binds to LPS. What we actually found as well is that it mostly binds to and depletes bacteria that encode hexaacylated LPS. So it actually doesn't deplete pentaacylated LPS producing bacteria in the gut. So what this is actually a quite targeted antibiotic for gamma proteobacteria or hexaacylated LPS producing bacteria. We also went in with a small molecule inhibitor of the receptor for LPS on the host, which is toll-like receptor 4. And what we found is that if we either deplete the bacteria that are producing hexaacylated LPS, or we block the host receptor for LPS that is responsible for immune activation, we significantly abrogate the ability of anti-PD-1 to improve anti-tumor immune response. And that's when we look at the immune cells that are able to infiltrate the tumors in these mice. So again, effector cells that are commonly associated with a robust anti-tumor immune response, interferon gamma producing CD8 and TNF, alpha producing CD8 T cells, are significantly increased in response to anti-PD-1, so the immunotherapy, the immune checkpoint blockade. However, when the mice are treated with this small molecule inhibitor TLR4 just during the immunotherapy, so they just start receiving this at the same time that they get anti-PD-1, or the mice are pretreated with something that depletes their hexaacylated LPS bacteria from the gut microbiome, we again completely lose this immune infiltration into the tumor. Importantly, if we add it back, we get improvement. So just by supplying hexaacylated LPS in the drinking water of mice, we were able to significantly increase responses to anti-PD-1. So I'm not showing the data here today, but actually if we give the LPS by itself, it doesn't have any effect on tumor growth. So it's in combination with anti-PD-1 that we see an improvement. So here you can see if we just give the pentaacylated LPS, which again we know doesn't robustly activate immune activation or NF-kappa B activation downstream of TLR4, we don't see any change in anti-PD-1 efficacy. But if we give oral hexaacylated LPS, we improve anti-tumor immune responses. And again, this is seen here in the abundance of different T cell subsets that are infiltrating the tumor. So with that, hopefully I've been able to give you just a little bit of a snapshot of how we are examining things. So by taking an observation in the microbiome in human patients, we were able to go into a mouse model, take the metabolite, take the function of the bacteria themselves, and examine the mechanism in an animal model and see that a microbiome signature of response to immunotherapy is identifying a microbial metabolite that can actually enhance the efficacy of the anti-PD-1. And then I hope that, I've been able to convince you that if we examine the functions of the microbiome rather than just the taxonomy, this actually leads us to important physiological mechanisms that potentially could be clinically tractable and important in understanding host immunity. And with that, I will I try to call people out as we go along, but I will thank everyone in my lab, which has grown substantially recently. I'm very proud of all of them. These guys with stars are alumni and have now moved on to other things. But also I want to thank everyone in CITIID and collaborators, especially collaborators at Memorial Sloan Kettering and Trevor Lawley, with whom we collaborated quite a lot in the beginning of getting the lab started.